Welcome to Third Eye Weekly. With us are the members of the Pieris Project, a citizen science investigation into the effects of climate change on biology, specifically on the cabbage white butterfly, and how the species can help us understand how other organisms will respond to climate change. Thank you for joining us.
Thanks for having us.
All right, so I understand that this is a way that you guys can actually have citizens participate in some of the scientific experiments. Tell us a little bit about the Backyard Genomics Kit and how engaging with the public makes for better science.
We might let Matt take.
Okay. Yeah, so part of what we're trying to do is collect butterflies in order to answer real scientific questions. And so part of what this genomics kit does is actually allows people who may not have access or equipment to catch butterflies to be able to do that, and then also learn about what's in their backyard, and send those samples to us to.
It's kind of like a little starter kit so people can get involved, learn about the diversity in their backyard.
So what's particularly unique about the white cabbage butterfly that makes it an ideal specimen for learning about climate change effects on organisms?
There are a number of reasons why we chose the cabbage white butterfly. One is that I would argue, it's likely the most abundant and widespread butterfly on the planet. So it's believed to originate from Europe and then spread across into Asia about 2,000 years ago. In the last 200 years, it spread to North America, including Hawaii, then also Australia, New Zealand, and other parts of the world. So it's everywhere, so lots of people can get involved. It's also quite abundant, so when we take a few of them to learn about them, we're not going to have a detrimental impact. So in terms of conservation, a lot of times we think about rare species, wanting to understand how they're doing. But they're usually very vulnerable to collecting individuals from this population. So by using a species that's doing quite well, we can learn about how it's adapting to changes in the environment and then apply that knowledge to other species where we couldn't do that. The nice thing about working with invasive species, so the cabbage white is invasive in many parts of the world. Invasive is too strong a word, it's not native to other parts of the world. So because of that, it's created what's called a natural experiment, where it's likely, as it's spread across the world, it's adapted to the different environments that it encountered. And because of that, we can use it to learn about how genome and other parts change right across the world.
You guys are looking also at the phenotype and the genotype, right? So looking at the changes of physically the butterfly changes in different environments, and then also genetic sequence of it.
Mm-hmm. So maybe Anne can talk a little bit about the phenotypic things that we're measuring, and then I can talk a little bit about the genomics.
Yeah, so one thing that we're really interested in regarding the phenotype of the butterflies is the pigmentation that they have in their wings. So they have a white pigment, hence the cabbage white name, and that pigment is extremely nitrogen rich. And so we're looking at not only how climate change is affecting that phenotype, but more just how any human-induced environmental change is affecting that phenotype. So specifically, we're looking at land use change and how the change into more agricultural land use has been affecting the pigment in the butterfly wing.
Excellent. So I'm curious, what inspired all of you to start this project, and what are some of your ultimate goals?
So the history of the Pieris Project. So I've been interested in citizen science for a long time now. I was fortunate enough to work with some people that have been doing some amazing things in citizen science. Inspired me, but also gave me a little bit of the skills and tools necessary to feel comfortable with creating a citizen science project myself. So I was in the process of launching the citizen science project, and then found out that Anne was also interested in having samples of the cabbage white from all over the U.S. And so we kind of joined forces, and then have been expanding out to other researchers to take on other things like uh, outreach with schools and other things that we could talk about. But so it started that way, and then it expanded from there. I would say the big primary goal of the project is to create one of the most amazing data sets for a single species. So it's ambitious. We want long-term data, so we'd like this project to go on for hopefully the rest of our careers and lives. And then also have it range wide. So this butterfly lives everywhere, have collections from all across the world, and have these collections at the population level. Sometimes in science, we only have samples of a few individuals at one location. But to really understand how species are adapting or evolving to changes, we actually need samples from many different populations. And so our goal is to have a few butterflies from as many places as it's so that's our grand goal and vision. And then with that, we'll answer a number of research questions. I should say that in science, we're often lacking long-term data sets. They just don't exist, so it's really difficult to answer questions on about how species are changing through time.
So how many butterflies have you guys collected so far, and what is the hypothesis you guys have for the data you will find from all these?
We have over 1,200 this last season. We got off to a late start. We started in, I think it was late June. Um, and as you guys live in New York, you know that there's not too much longer in the season. Although this butterfly does come out, it's one of the first that comes out in the season. We've already seen it here in Indiana, so it's probably going to be more soon. And then it hangs around for most of the year too, so that's also a cool thing about this butterfly is it's one of the first out, one of the last to go in for the winter. But we have about 1,200. We hope to do better than that this year. We really would like to have butterflies collected from all 50 states. I think we got 26 states last year. We have a number of other countries where we have butterflies: Australia, New Zealand, Japan, Finland, Portugal, Spain, the Czech Republic, a few others that I'm forgetting. But basically expand beyond that this coming spring. And then the question was about hypothesis for the genetics. Is that it?
Yeah, what's the hypothesis you guys have for what will define how these butterflies adapt to the environment?
Okay, so one of them is about looking at how they adapt to changes. So basically, we would be looking at associations between genetic markers and environmental variables. So that could be with what Anne was talking about. We could look at nitrogen or a proxy for nitrogen, which is how much agriculture is in an area, and then how much that pigment is there. So we can do that with traits and environmental variables, but we can also do that with genetic markers. So the variants of the genes inside them might be correlated with particular environmental variables. So we might find in really warm locations they have a lot more of this particular variant of the gene than they do in cold regions. And because if we have collections from all across the world or many different locations, we can actually replicate that. So it could just be by chance that we find, if we look only in New York, some cooler and warmer places, we find differences, but it just might be by chance. So to be able to do that in multiple different places really helps us to determine what is causation versus correlation. But we're also, there's one other thing that we're doing, in addition to just looking for those associations between the genes and different environmental variables, is also to reconstruct the invasion history. So with genetics, we can actually, it's kind of like forensics in a crime scene. We can try to figure out where each butterfly came from. So we believe they came from Europe, but we actually don't know exactly where in Europe they came. Could have even came from Asia. So we can use the genetic markers inside them to map back to the locations in Europe and Asia to see which is the best match. And see if there's been multiple interactions. So what happens a lot of times with invasive species is that it won't be just one time where that organism was transferred over to a new location. It'll happen multiple times. And so we want to see whether this butterfly was introduced multiple times with multiple different variables. That can help us understand why it was successful in spreading.
are there other insects that you'd love to examine for future studies, or are you going to stick to mostly butterflies?
Um, Definitely other organisms. Doing just one organism is a lot of work, though. So running a citizen science project is a lot of work, especially if you want to do it well. So we're trying to add more material online. We want to have a quick turnaround time, so taking the butterflies, processing them, sequencing them, getting the information back to people. We're working on that right now. We're actually sequencing some butterflies, hopefully to have the DNA soon. We're trying to get just at least one butterfly from every person that sent one in, so that you can kind of, each person can see how their butterfly compares to those that other people have sent in. So doing all of that takes a lot of work. At some point, we would love to expand, and we'll probably expand to other species. But this was kind of to get things started. Um, I have a fondness for beetles. Why I was interested in some other organisms, and that with mosquitoes. So at some point we may expand, but it's just a lot of work to do, even just butterflies. That and trying to graduate. I should say that we're all graduate students, so none of us actually has a PhD yet. We're all working towards a PhD. So getting a PhD is a lot of work, and this is a side project that we're working on.
I hear, are you guys in science jail towards the end of PhD where you're just kind of locked up and try and defend your thesis?
No.
Are you guys not at that point yet? Are you almost done?
I'm sorry. I'm just a second-year grad student, so I have a few more years, probably at least 3 more years to keep going through. And I'm fortunate enough that this project actually does help me collect some of the data that I will use for my dissertation. So this is more of a primary aspect of my project, whereas the rest of the group this is just a side project for them.
Yeah. I am trying to graduate in August, so it is crunch time. Yes, it's from butterfly collecting season. So it's tough to have the summer focused on writing my dissertation when I'd like to be catching butterflies, emailing the people that write in and tell us all the cool things that they're seeing in their backyard.
So lastly, what is the importance of this study you guys feel on society and in science overall?
I think citizen science in general is really important because it allows people to participate in science. It used to be that way, that in the beginning of science, that a lot of non-professional scientists were involved in science, and then it slowly became pretty much only science was done in academia or in the private sector. So I think it's really important to go back to how it was, where anyone that's interested in science can participate, and not just doing like a mock experiment, but actually being involved in a research question that none of us know the answer to. I think that's really exciting, and I find that a lot of people find that really engaging and want to help and want to participate. So I think citizen science is important for that. It's also just amazing when we collaborate with the public, the things that we can do. And you can imagine we'd all like to travel around the world to collect these butterflies, but we definitely can't afford it in terms of time and money. So having people that can help us by just going in the backyard and sending a sample is huge. It means that we can now have more butterflies, but also from more locations than we've ever think possible. So it's very efficient. Saves a lot of money.
Yeah, I would also say that with citizen science, I think a lot of times people who aren't scientists sort of feel that science is this abstract thing. And so with citizen science, it sort of allows them to be involved in the process and recognize if they're running an experiment, and also for kids, what we plan on doing is having some sort of educational component. So kids or adults who learn about what people are thinking about. So also our people who are scientists learn about science and also see what scientists are doing.
So then, kind of on my aspect, if you're thinking about more of like a hypothesis-driven research project, so I'm really interested in finding, or our group is really interested in finding how this human-induced environmental change is affecting the evolution of these pigments in the wings. And so we would hope to find, or maybe not find, a lack of any evidence would, be helpful. But we would just want to know how those how humans are affecting these traits.
So how humans are affecting the climate and therefore affecting the traits, or how humans, their environment in general, are affecting the traits of the butterfly?
Right, just how humans in general with land use change and the input of a lot of nutritional aspects like nitrogen through fertilizer and how that's affecting these bugs.
Yeah, I would say land use, climate, all these things are all human activities are influencing climate, land use, movement of species. We know that we're doing that. We know it's having a big impact, but it's hard for us to predict how these changes are impacting biodiversity. There's some general responses, like range shift. Species are shifting their range, particularly in response to climate, or their phenology, that is the timing of when they're doing what they're doing. We know that very well. I'm sure many people have heard or seen about trees that are leafing out earlier in the season as it's getting warmer, or birds migrating earlier than they used to. So we know in general that species are going to shift their range. They're going to shift their timing. But the question is, are they going to adapt or evolve to these changes, in response to these changes? And that we don't actually know very well. So part of our project, the goal is to learn how species adapt to climate change or other changes, land use changes. And in order to do that, we have to have data sets that allow us to answer those questions. So long-term data over a broad spatial area.
All right. Well, those are all the questions we have today. Thank you guys so much for taking the time to speak with us.
We should say that anyone that wants to help is welcome to join. It helps to have a butterfly net, but if you're a ninja and you can catch them, that's also a possibility, or a jar. And then on our website, we have ways that people can get involved into either building their own butterfly net or buying one online. And then how you can make a little envelope to store your butterfly.
We'll put the link up on our site. Victor. Now that the weather's getting warmer, I'm sure.
Yeah, they should be out soon.
All right. Well, thank you guys so much. We appreciate it. We'll send you guys a link when everything's all edited and together.
Great.
Cool. Thank you for having us.
Thanks.
Thank you.
Have a great day.
You too.